Ms. Mann, we're having some audio issues, and so I'm going to ask you to get close to the microphone, with your face close to the microphone when you answer so it projects, okay? Thank you so much.
Ms. Mann, could you introduce yourself to the jury, please?
Yes, sir. Hello, my name is Lori Mann. I'm a forensic biologist at the Bureau of Alcohol, Tobacco, Firearms, and Explosives.
How long have you been doing that?
I've been a forensic biologist since 2013. I've been with the ATF since 2017.
Could you explain to the jury what forensic biology is?
Yes, forensic biology is the examination of evidence in a crime to determine if there are biological materials present. We examine those materials for the possible presence of DNA evidence and make comparisons between that evidence and known reference samples.
What educational background do you have?
I have a Bachelor's of Science in Biochemistry and Molecular Biology, as well as a Master's of Forensic Science in Forensic Biology specifically. Both of those are from the Oklahoma State University.
And how many examinations were the Bureau of Alcohol, Tobacco, and Firearms have done?
Examinations specifically or cases as a whole?
How about both?
I would say between 20 and 50 cases a year. Each case may have an average of 10 evidence samples. It's hard to have an exact number, I'm sorry.
A bunch.
It's quite a few, yes.
Have you ever been called on to testify as an expert in the courts of the United States, as an expert in forensic biology?
Yes, I've testified about 10 times as a forensic biologist.
Okay, this time we tender Lori Mann as an expert in forensic biology.
She's so qualified.
Ms. Mann, could you explain to the jury what DNA is?
Yes, DNA is a molecule within the cells of our bodies, and it codes essentially for the way that we develop as humans. You inherit half of your DNA from your biological mother and half from your biological father.
So what is it made of? DNA. Dio something.
Deoxyribonucleic acid is the scientific term, but yeah, we call it DNA for short. It is made of different proteins, essentially, sugar phosphates.
Do all humans have DNA?
Yes, all humans have DNA.
How does the science of DNA work to determine a person as touches?
So what you're asking about specifically there is the activity of deposition, the activity of how DNA got onto an item, and our testing cannot allow us to make that determination.
So how do you determine if DNA is present?
So the DNA process in the lab is pretty straightforward. We apply pretty much the same process to all of the evidence that we get. When I receive an item, I will collect samples from various parts of that item, depending on how I think it may have been handled, or if I see possibly blood present, things like that. So I'll collect those samples, typically with a swab. I use a wet swab followed by a dry swab, and that's the DNA sample that I take forward for testing. So that sample goes through several steps of testing in the laboratory, and at the end, I hope that I have a good DNA profile that I can then use to compare to a known reference sample from an individual.
Were you asked to do the DNA test as relates to this case?
Yes, I was.
The issue has been previously admitted as State's Exhibit number 13 as a proposed mass treatment location shooting at QT gas station in July 10, 2022. Did you come into contact with the contents of State's 13?
Yes, I can see my initials on this package.
I'm going to show you as previously admitted into evidence of Stage 42, which is it buckle or bugle?
Buckle is how I pronounce it.
You're the expert. So let's go with buckle. Stage 42 previously admitted as the buckle swab of Josiah Coleman. Do you have contact with that?
This is not the swab you're referencing. This is a swab that I actually collected from the mask during my processing.
Okay. And when you collected that swab, did you compare the swab to the buckle of Josiah?
Yes, I did make that comparison in this case.
certainly.
Space 42, what was that again? Space 42?
So when I processed the mask, I noticed a significant area of red staining. I tested that area for the possible presence of blood, and it was positive. From that area, I took these swabs, and then I sampled a part of these swabs for my testing process because blood typically has a high amount of DNA, a high quantity of DNA, so I don't need to use my whole sample. So this is essentially what was left of that swab after I performed my testing.
Just 1 second.
Exhibit 65. Ms. Drake? Ms. Drake?
There's still not been any
Okay.
testing on how that relates to.
Okay, that's your objection. Okay.
The issue is state 65, which is the doctor swab, which is
Yes, sir.
You recognize state 65?
Yes, I can see my signature here on this paper chain of custody.
And what is the contents of state 65 as it relates to your investigation?
This is a known reference sample from Josiah Hughley.
And were you able, and when you received space 13 and space 65, were they sealed?
Yes. So the packages were sealed, and then after I did my examination, I added my own seal onto it from where I opened it.
Now, when you received it in a sealed condition, you had the seal been tampered with?
I don't recall if there was tampering. That would be something I would record in my notes.
Did you record that in this?
I'm sorry, sir. I genuinely don't recall. It does appear to be sealed appropriately at this time.
And this, had the sample been tampered with, is that something you would have indicated in your report?
Absolutely. Seals are very critical for maintaining chain of custody, and so if there's anything unusual in the seal, if it's not sealed properly, that is something that I note, and then I also call the special agent or the investigating agent who submitted the evidence to me, and I let them know that it wasn't properly sealed.
In this case, did you notate any tampering at all in your notes?
Not that I recall.
I'm going to show you, and I'm sorry if this place states. Here it is. States 81. Did you generate a report as it relates to this case?
Yes, sir. I generated 2 reports in this case.
I'm going to refer to states 81. Is that report as related to this case?
Yes, this is the second report that I generated.
And in that report, does it contain the contents of the examination you did between states 13 and 14?
States 13 being the mask. Yes, it does.
Did you notate any tampering at all?
So that would be in the notes that I take as a process of opening the evidence, which is not contained in the report. But again, I do not recall if it was open or unsealed as I received it. Even if the interior packaging was open and or unsealed, typically there are multiple layers of packaging, and so sometimes the exterior layers will be sealed, and that seal will be sufficient for the interior layers. I apologize, I don't want to misremember.
States 13, the COVID mask. You did the DNA testing on that. What was the result of the test?
Yes, sir. So I collected several samples from this mask. Is there one in particular you would like me to address, or shall I go through all of them? The mask specifically?
Yes, ma'am. The mask that states 13, and that is in relation to the testing done on buccal swab.
I'm going to reject, may I ask one question, and I can give my objection
Okay.
for the objection? You said that all of those packages are now sealed. When you were answering a question of Mr. Bellman, you said they're also properly by KR now. So all those packages are sealed. Would that be correct
So when I'm looking at the seal, what I'm looking for is tamper-evident tape, which I can see that these all have covering, or this one specifically that I'm holding, has covering the openings. I'm looking for initials and date on that tape, so across the tape and onto the package. So it is intact there. I see initials on this tamper-evident tape. I see a seal in there, which I think is probably the seal that I applied because that looks like our evidence tape from what I can see. That appears sealed, and then I see here, this is the seal that I created when I made this exhibit. Looks like there's some tape that was removed there, but it doesn't appear to be an opening, so this also appears appropriately sealed.
Your Honor, I'm going to repeat my objection. The chain of custody has not been established. She has just testified that even when something is open, it can be concealed. She herself did not remember if there was anything wrong with the seals when the sample was given to her for comparison. And we don't know who took it there, how many people touched it, was it ever open, was it ever resealed? So I would object to the chain of custody.
Each one of those issues goes to weight and visibility. Crime scene technician said she sealed it. This man said she generally sees it sealed, but it never been there. That's what it means.
I agree it goes to weight, and I'm gonna overrule the objection.
Let's get to the result. What was the result?
Yes, sir. I collected several samples from this mask, so I'll just go through each one and describe what I've collected. Exhibit 1.1, which I collected again, was swabs of the staining that I saw. Then I took a sample of that. That became Exhibit 1.1.1. So that is a cutting of a swab of reddish-brown stain from the mask. That's the first sample I took. Sample 1.2 is swabs that I collected from an area of fluorescence on the blue side of the mask. So this is sort of a standard mask that we all saw during COVID, those disposable blue and white masks. So I examined it for what we call fluorescence, which is an indication to me of a possible biological fluid such as sweat or saliva. And so I collected specifically from that area of fluorescence on the blue side of the mask. Exhibit 1.4 is a swab of the white side of the mask, including an area of fluorescence. I see that 1.3 is not in this report. That's because this is a lighter report, and that one was not suitable for comparison, so we leave it out of later reports. Exhibit 1.3 was swabs of the remaining area of the blue side of the mask, so I swabbed the area of fluorescence, and then I swabbed sort of everywhere else, and that one isn't in this report, again, because I wouldn't have made comparisons to it. And then finally, I cut open the mask. So inside the mask, there's essentially like a filter. So it's sort of coarse fabric type paper. And I noticed that that also had fluorescence on it. And it's important that it was on the interior because the exterior, if you'll remember, had possible blood staining. And so when there's blood staining present, blood can tend to flake off and get all over everything. And so I'm looking for areas that were protected from that possible blood staining. And so by cutting it open and getting that protected area on the inside, that's sort of a way that I can reduce the number of contributors to a DNA sample. So I took a cutting from the area of fluorescence on the interior filter of the mask, and that was my final sample.
When you did the comparisons, what was
So I made comparisons to each of the known reference samples that I received in this case.
let's focus on the reference sample just prior to the differences.
Yes, sir. So it's important to describe how we reach our conclusions. Can I? Okay. So when we're making a comparison to a known reference sample, the type of calculation that we do is called a likelihood ratio. And so this is a way of describing the weight of the DNA evidence when we make that comparison. And so a likelihood ratio compares two different explanations for given observation. So our observation here is the final DNA profile that I developed. And so we're trying to think about what are two ways that we could have seen this DNA profile. So we compare those two hypotheses. So in general, we're comparing the possibility that a known individual contributed or that any other known and unrelated individual in the population contributed to the sample. So I made that comparison with Josiah Hughley for Exhibit 1.1.1 and found support for exclusion of Josiah Hughley in that sample. So it's important to note that the likelihood ratio will provide us a weight. So that number can be a large number, that number can be a small number. And the size of that number is what allows us to reach a conclusion. So we do have 5 different conclusions that we can reach. We can generate a conclusion of support for inclusion of an individual in a sample. We can say there's limited support for inclusion in a sample. We can say there's limited support for exclusion of a sample, and we can say an individual is excluded. And then there's a 5th one that's not in the report because we don't see it too often, and it's just inconclusive, which just means the ratio doesn't tell us very much. So from the cutting of the reddish-brown stain on the mask, the comparison was that Josiah Hughley was excluded as a possible contributor to that sample. For the swabs of the fluorescence on the blue side of the mask, there was support for the inclusion of Josiah Hughley as a possible contributor to the major component of that sample. And a major component is just a part of that sample that we've determined is suitable for comparison. So the major component may mean that some individuals contributed more to the DNA sample than other contributors. And so we're only making comparisons to those major component contributors. Then I have Exhibit 1.4, the swab of the white side of the mask, including possible areas of fluorescence. There was support for the inclusion of Josiah Hughley as a possible contributor to that sample. That was just a single male contributor in that sample, so not a mixture. And then same with Exhibit 1.5, again, just a male DNA profile consistent with a single contributor. And this was the cutting from the interior filter of the mask. And there was also support for the inclusion of Josiah Hughley as a possible contributor to that sample.
You previously mentioned likelihood ratios. What's the likelihood ratio?
Yes, so I list the statistical calculations in my report for each comparison that we make. So for the comparison to Exhibits 1.2, which is the area of fluorescence on the blue side of the mask, that's grouped with a different exhibit that we haven't mentioned, so I'll leave that one out for now. This mixture, essentially the sample resulted in a mixture of 3 individuals. And again, we're not making comparison to all 3 individuals, just a major component to the profile. So I mentioned we're talking about what are 2 possible ways or 2 possible explanations for seeing this sample. So those 2 possible explanations that we compared here were that the DNA originated from Josiah Hughley as a contributor to the major component, along with 2 other individuals, or did the DNA originate from 3 unknown, unrelated individuals? So we made a comparison of those 2 possibilities. And so the result here was that the DNA profile seen in Exhibit 1.2 is at least 1 trillion times more likely if it originated from Josiah Hughley as a contributor to that major component, along with 2 unrelated, unknown individuals, than if it originated from 3 unknown, unrelated individuals.
I want to make sure I heard you right. That's 1 trillion for TR.
Yes, sir, 1 trillion.
Thank you. Now let's move away from that. I just want to. Are you familiar with principles of degradation and contamination that relates to DNA?
Yes, sir.
Could you explain what degradation is?
Yes.
Well,
So.
can I just make it a little clearer since you're qualified as an engineer. If I have a pound of chicken right here, and it sits there for 10, 100, 1,000 years, is it possible that it would degrade to such an extent to not get a DNA, insufficient DNA? Say it's chicken. Is that a possibility?
I'm sorry, I am very unfamiliar with DNA testing
Okay.
for chicken.
Okay, but I mean, is it possible that a living substance, DNA, can degrade to such an extent that you cannot tell what it is?
So with the testing that we use, yes, it's possible that samples can become too degraded that we cannot use them to compare to known reference samples.
So in DNA world, trying to keep it simple. Even if it degrades, it'll never morph into beef or turkey as far as testing is concerned. Is that correct?
That's correct. So degradation is essentially where the DNA that's present breaks down due to, it could be any of a number of different factors. So what we see from a degraded sample is essentially a loss of data. It doesn't change the data in any way. It just reduces the overall amount of data that we see. So factors that can degrade a sample can include exposure to heat or sunlight or bacteria, things like that.
And as far as contamination, just for my part, keep it simple. If I have a pound of chicken and a pound of beef, and a pound of beef touches the chicken, theoretically, if I take a DNA sample at a particular spot, it could test for chicken, it could be contaminated, it could test for beef, might even contaminate for. Is that about right?
So just drawing on your analogy, if two evidence samples come in contact with each other, yes, DNA transfer can occur through that contact.
Okay, so, but the chicken and the beef, even if it's contaminated, will never test as turkey, correct?
So that's also correct. I think I see what you're trying to say here. We're not going to create a third DNA profile. Exactly. It's not going to morph into something else or change. Yes.
And when you're talking about a likelihood ratio of 1 in a trillion, that kind of excludes that there was any decontamination or contamination of data. Is that fair to say?
Hunter, I'm going to attach, you can't testify to contamination because we don't have everybody here who actually says you don't know what they may or may not have done to it, so it's speculation.
1 in a trillion. In other words, the sample that she tested, you would have had to test it as opposed to actually. That's what Lisa's doing.
I'm going to overrule the objection. She's an expert.
Yes, thank you. So, chicken and beef touch, even if those are contaminated, it'll never come out as a third.
Correct. Two DNA samples touching each other will not create a third different DNA sample.
In the sample that you tested, you didn't see any degradation. You were able to get results of degradation.
That doesn't mean there was no degradation. We do often see small amounts of degradation in evidence samples. It's not uncommon. For things that are extremely or highly degraded, we may not get all data present. Sometimes we may get what we call a partial profile, which means some of the data isn't there. And if the partial profile is good enough, we may be able to still make comparisons to it. But if a sample is highly degraded such that there's little DNA remaining, then we may not be able to make comparisons to it.
But your finding was the likelihood ratio is 1 in a trillion.
That's correct. That was the result of this likelihood ratio.
Thank you.
Mr. Lewis.
Yes. Good morning.
If not, my name is Lawrence Lewis. If I ask you a question you don't understand, you just let me know. It's lawyers asking science questions, so it may be a little awkward. I hope it's not, but if it is, don't be shy
Yes, sir.
in correcting me. So let me start here. In July of 2022, for whom did you work?
I worked for the ATF at that time.
Give it to me again.
I worked for the ATF, that's the Bureau of Alcohol, Tobacco, Firearms, and Explosives.
And in July of 2022, how long had you worked for them?
Since late 2017.
So it's been about 5 years.
Yes, sir.
And your job title was what?
Forensic biologist.
That's what I thought. I got you. Are you still working for the agency?
Yes, sir.
I'm assuming working for ATF, then you've been qualified to testify in federal courts. Is that right?
I have testified in federal court, but primarily state courts.
I got you. The prosecutor gave you an example about degradation. State prosecutor here, they gave you an example about degradation over time. Is that right? Did he give you that example?
We did discuss degradation, yes.
Okay, and in my mind, it's not necessarily degradation over time, but it's degradation as a result of exposure to bacteria. Do I have that right, or do I have that wrong?
So there are many factors that can degrade a DNA sample. Bacterial exposure is one of them. Also, I mentioned sunlight, heat, things like that will break down, and the bacteria specifically essentially chews up the DNA that's there, and so we can lose some of the sample to degradation in that way.
Does blood degrade by evaporating?
No.
Okay, so it's not just time. Something else has to occur during the time in order for that blood to degrade. Is that fair to say?
Yes, so we can think of it as the application of those degrading factors for an extended period of time that could increase the degradation.
Right. Have you ever heard of digital DNA?
I'm not familiar with that term, no.
All right, thank you very much. No further questions.
Mr. Clegg.
My name is Tom Clegg, and I represent Mr. Miles Collins. In addition to the buccal swabs, that for years I had couldn't pronounce your swabs.
That's fine,
Glad
too.
you've corrected me. You also received buccal swabs from Miles Collins. What were identified as coming from Miles Collins, right?
That's correct.
And you compared them with the samples taken from the mask, right?
Yes, I did.
And you excluded Mr. Collins as a potential contributor to any DNA that was found on the mask, right?
Yes, so for each of those 4 samples that I made comparisons to on the mask, Miles Collins was excluded as a contributor to the sample.
All I needed to know. Thank you very much.
Yes, sir.
Great.
Now, you do not know Josiah Hughes, correct?
That's correct.
And the only reason why you were labeling the sample that you tested and compared as Josiah Hughes is the package that you received from another agency.
So typically when we receive things, it is from our own agents. So it does have to go through our agents and our agency to be submitted to the lab.
Where do they
I can see on this package that there is a Gwinnett County Police Department label. I can't speak to the exact transfer that occurred as I was not present at that time.
But the reason why you're saying Josiah Hughley is that's what the package says.
That's correct. Yes, that's how it was submitted to me.
That's all.
Can you redirect?
Yeah, Your Honor, we asked that you release this.
No objection.
All right, Ms. Mann, thank you for testifying.